Hi everyone, welcome to my first seminar presentations. My name is Minh Quan, or you can call me Harry. I'm a second semester master's student in the Department of Biomedical Engineering, and I'm currently working in Lab 510 of Professor Xiao in Bioscience Buildings. Here's my content for my presentation today. First, I will do some self-introduction and then my education background. After that, we introduce my technique and the domain in knowledge. The technique name is CRISPR-Cas9 and its application in biomedical and their novelty. First, I will do my self-introduction. I was born and raised in Ho Chi Minh City, which is one of the largest cities in Vietnam. Vietnam is a Southeast Asia country. We are neighbor to China, Laos, Thailand, and Cambodia. And my birthday was 11 November 2001. And one of my favorite activities is running and food explore, which is I very enjoy in Taiwan. Is the food, the night market culture. I want to talk a little bit about my hometown, Ho Chi Minh City, or the Vietnamese local they prefer calling the name Saigon. When you go to Saigon, it's in the southern of Vietnam, and you can find some very interesting and famous food in the world, Vietnamese food in the world, like pho, Vietnamese pho, or banh mi, or Vietnamese coffee. And you can visit some very beautiful place, like this. I have an image about the Notre Dame of Saigon. It's in the center of the Ho Chi Minh City. And for my education background, from 2019 and to 2023, I was studying in Industrial University of Ho Chi Minh City, and I was major in chemical engineering. And my thesis and my research field back then is what material and application for wastewater treatment. And from 2024 to 2026, I was pursuing my master degrees in Trung Nguyen Christian University in Department of Biomedical Engineering. And currently my research field is the wound healing and study with the animal model, specifically is zebrafish. So next is my introduction about the technique or technology. So today I will select CRISPR-Cas9 to present to all of you. So CRISPR-Cas9 is a groundbreaking genome editing technology that allows scientists to make precise change in the DNA of living organisms. So what does CRISPR stand for? The CRISPR stands for clustered regularly into space, short, palindromic repeats. This is a natural defense mechanism found in bacteria, which they use to fend off virus by cutting their DNA. And scientists have adapted the system for use in genetic engineering. And I have summarized some of CRISPR-Cas9 overview, and this is the overview of the technology. So CRISPR-Cas9 is a revolutionary genome editing tool derived from a bacterial immune system, and it allows scientists to precisely cut and modify DNA at specific location. And originally discovered in bacteria, CRISPR-Cas9 would adapt for gene editing in 2012 to now, and it's enable researchers to delete, insert, alter genetic material in highly targeted manners. And from 2012 to now, it's widely used in genetic research, disease modeling, and developing new therapy. And why a lot of the researchers select CRISPR-Cas9 to study? Because this technique or technology is simplicity, low cost, and versatility make it a game-changing technology in biomedical research. So I want to give you a brief history of the technology. So in the 2000s, scientists studying bacteria discovered that CRISPR sequences and Cas9 protein, and they are realizing that this system could target and cut specific DNA sequencing, like I mentioned before. But the breakthrough came only when they understood that this bacteria defense mechanism could be repurposed for precise genome editing in any organism, not just bacteria anymore. And the application of the genome editing in 2012 and 2013, and researchers like Jennifer Donna and Emmanuel Charpentier adapt CRISPR-Cas9 into a tool that can program to target specific genes in a wide range of organisms, including humans. And they realized that by designing a specific guide RNA, they could direct Cas9 to cut DNA at any desired location in the genome, open the door to precise genetic modification. And by this discovery, Jennifer Doudna and Emmanuel Charpentier has won the Nobel Prize in Chemistry. And I have a papers right here. The name is Nobel Prize for Chemistry Award to scientists who discovered CRISPR gene editing tool for rewriting the code of life. So how did this work? So for understanding the CRISPR-Cas9 technology mechanism, first I want to give you a brief introduction about two things. First is the guide RNA or gRNA. It's a short RNA sequence designed to match the target DNA. It's direct the Cas9 enzyme to the specific location in the genome where the cut will be made. It's like a guidance. And second is the Cas9 enzyme. You can imagine Cas9 enzyme as a scissor. So this molecular scissor that cut the DNA at the target site. So Cas9 is the part of CRISPR system that facilitates precise genome editing. And you can use this image here to imagine how the CRISPR-Cas9 work. So the CRISPR-Cas9 enable precise gene editing by using a guide RNA to direct the Cas9 enzyme to cut and modify specific DNA sequences, and allowing for target genetic change. The gRNA guide Cas9 to a specific DNA sequence, and Cas9 create a double-strand break in the DNA in this location. And after that, the cell prepare the cut, either the non-homologous end joining or NHEJ, which is often introduced a small mutation, or by homology direct repair or HDR, if a DNA template is provided, allowing precise genetic modification. I have prepared a short video clip here to summarize all of the knowledge about the concept and the mechanism of the CRISPR-Cas9 technology in genome editing.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then, they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care, or even cure lifelong inherited diseases.
So through this video, I think it's very easy to understand the concept and simple mechanism of the CRISPR-Cas9 technology in the genome editing, and some of it application in biomedical research. And to make it more clear, I will give you the CRISPR-Cas9 mechanism. So through the video clip and through the concept that I have presented in the previous slide. So first, the scientists will design a guide RNA or gRNA. It's design engineering to match the target DNA sequence that you want to edit. And second is to binding the tRNA to target DNA. The tRNA bind to the complementary sequence in the DNA, guiding the Cas9 enzyme as a scissor to that specific location, and the Cas9 will cut the DNA and creating a double-strand break at the targeted site in the DNA. And after that, as the DNA repair, the cell attempt to repair the break through one of two pathways. First is a non-homologous end joining, are often introduced in small mutation that can disrupt the change or knockout. And the second is the homology direct repair (HDR). Is used as provide DNA template to make precise edit, like inserting a new change or knock-in. And the final result is the target gene is either disrupt or precisely modified, depending on the repair pathway that the scientists want it to be. So after understanding the mechanism of the CRISPR-Cas9 technology, so what was its application in biomedical? So some of application like gene therapy, disease modeling, cancer research, drug development, stem cell research, or antiviral therapy. And for gene therapy, CRISPR is used in research and clinical trial for treating genetic disease by correcting mutation at their source. And for example, it has shown that promise in treating disorder like sickle cell anemia or muscular dystrophy by fixing faulty genes. And for cancer research, scientists and researchers use CRISPR to identify genes involved in the cancer progression by knocking out specific genes in cancer cells and observing the effect. And that will help identify the new drug target to that cancer. And you can see in the image, the CRISPR-Cas9 technology is routed into three pathways. First, in the research, like chromatic interaction generating model, and therapy like knock-in, correction, knockout, or regulation, or drug development. And third is diagnostic. So application in like in some like diagnostics technology, and also for addition the CRISPR-Cas9 technology also application in agriculture. And CRISPR is being used in agriculture to create crops that are make resistant to disease, pest, or environmental stress, and that will enhancing food security. So for specific biomedical research application, I will briefly present papers that relating to zebrafish, which is the animal model I study in the lab. And the title of the paper is Evaluation of the CRISPR Gene Editing Tools in Zebrafish Model. And this paper is Q1 and impact factor is 3.5. So to make it more clear about the application of CRISPR-Cas9 technology in biomedical research, I only give you a brief describe of the papers. So this paper using the zebrafish as an animal model to study. So why they choosing the zebrafish? Because the zebrafish are flavor and gene function study, because of their rapid development, external fertilization, and the ability to produce many offspring. So the use of the CRISPR-Cas9 has enhanced the ability to create gene knockout, allowing for efficient targeting of multiple genes simultaneously. So for methodology and the experimental design, the study involved in microinjecting the zebrafish embryos with 50 different guide RNAs, targeting 14 genes to assess the accuracy of various CRISPR design tools. And the data collection is the researcher, they compare in vivo editing efficient with prediction from 8 commonly used gRNA design tools, and focusing on both on-target and off-target effect. So this is their graph representing their experimental design. And for this slide, it has summarized some of the results of the papers. Like the study involved 50 guide RNAs targeting the 14 genes in zebrafish embryos. And experimental results show that significant difference between the predict and actual editing efficiency, which an average efficiency of 57.6 across each RNA, but prediction often underestimate this value. And the researcher confirmed that a range of the CRISPR-induced insertion and deletion using the sequencing technique, and they find that while the tool like ICA and the TYDE provide like a correlated score, but they are consistently underestimate actual editing efficiency compared to the Illumina sequencing. And for the off-target mutation, based on the result, the analysis indicate that the lowest frequencies of off-target mutation is smaller than 1% in vivo, suggesting that while off-target effect does exist, they may not significantly impact the study using the stable mutant line. And for the control of the embryo analysis, the RNA sequence data from control embryos in check with Cas9 mRNA without the gRNA revealed that several hundred differential expressed genes, indicating that potential confounding effect from the microinjection process itself. And for the conclusion of the papers, so this will give like a future study, a future researcher. So this finding serve as a valuable resource for the zebrafish research community, and facing the need of the careful selection of the CRISPR design tool and consideration of the potential confounding factor from the microinjection. And this study called for further investigation into the implication of the microinjection effect and improving the prediction accuracy for the CRISPR application in the zebrafish and the biomedical research. And what is the novelty of the CRISPR-Cas9 technology? For example, like the precision oncology. So using the CRISPR to create a patient-specific cancer model for personalized drug screening and treatment strategies. And another example is like a gene drive, implements CRISPR gene derived to control mosquito population and combat vector-borne disease like malaria. So CRISPR-Cas9 technology have several novelty in biomedical research, and this is what I find the key novelty of the CRISPR-Cas9 technology. So first is the precision. The CRISPR can target specific sequencing in the genome with a great accuracy. The second is the versatility. It can be used in a wide variety of organism and cell type. For example, the zebrafish model, like I mentioned in the paper. And third one is the efficiency. The CRISPR is faster, cheaper than the previous genome editing method, making it accessible to more researchers and scientists around the world to study and do research about the genome editing. But however, the challenge remains, especially the concerning about the off-target effect. In the paper, they also concerning about this effect, and where the unintended part of the genome may be altered, but carefully experimental design can be a huge part to smaller change to cause this risk in your study or research. So what is the future of the CRISPR-Cas9 technology? So it is likely to be many years before the CRISPR-Cas9 in use routinely in humans. Much research is still focused on use on animal models or isolated human cells, with the aim of eventually we use the technique in the human to treat the disease in humans. And there's a lot of work focusing on eliminating the off-target effect, like I mentioned before, whereas the CRISPR-Cas9 system cut at a different genes to the one that intend to be edited. And that's with my self-introduction and my presentation about the CRISPR-Cas9 technology application in biomedical research. And finally, I want to thank you, Professor Ge, and fellow student, for listening to my presentation. Thank you so much.